Hi, and welcome to my presentation for the Astrobiology Pro Seminar. My name is Mariana Moore. Before I get into talking about my paper, I'm going to tell you all a short story about the Apollo 7 mission in 1968. So this was the first crewed flight in NASA Apollo's mission series, and 3 astronauts went into space, including Commander Wally Schirra. On the first day in space, Schirra got a really bad head cold, and by the second day in space, all of the 3 men were congested with the same head cold, and they were really miserable. They ran out of tissues and medication, and in space, there's not the gravitational environment like on Earth, so any fluid accumulating in the sinuses can't drain away properly. So a cold can become really serious. By the end of this 11-day mission, the astronauts were exhausted, uncomfortable, and frustrated. And I found this quite funny when I read about it, but they actually started arguing with mission control, and they refused to wear their pressurized helmets when re-entering into Earth's atmosphere, because they were worried that these pressurized helmets would cause them to burst an eardrum. So instead of wearing their helmets, they actually took pillows and cushions and whatever they could find and cushioned their heads on re-entry into Earth. And this was the last flight for any of these 3 astronauts. Now, don't get me wrong, overall it was a very successful mission, but this head cold caused a serious halt in the mission, and all of these astronauts really didn't have a good time up there. After this, NASA implemented a pre-flight quarantine, but still since then there have been numerous cases of cold outbreaks and even virus reactivations in space. And this brings me to the paper that I have chosen, and it is titled Spaceflight Virology. What do we know about viral threats in the spaceflight environment? This is a paper that was published in February of this year, and it is a short review paper. So the scientists haven't generated the data themselves, but they have compiled data from many different publications. What this paper is about is viruses in the space context, and also viruses in the microbiome of the ISS surfaces. They also talk about the influence of environmental factors related to space travel on viruses and on their hosts. And lastly, they talk about the control of viruses in space travel. So before I get into talking about the paper, let me first just explain what a virus is, so that we're all on the same page. So the first definition we had of a virus is a disease-causing particle that passed through a filter retains all known bacteria, so a contagious living fluid. And this first definition was in 1898. But since then, we have developed the electron microscope, and this has allowed for a much better understanding of what a virus is. So we know that all viruses replicate only inside the living cells of an organism, be that a human cell or a bacteria cell. All viruses have an extracellular virion state, and a virion is simply the complete infective form of a virus outside of the host cell. All viruses also have an external protein capsid, so this is just like a protein layer encapsulating the nucleic acid inside. And some viruses also have a lipid outer layer called an envelope. All viruses also contain genomic nucleic acid. So I've just included this image here, if I just can zoom in. So like I said, all viruses contain genomic nucleic acid, and this can be DNA or it can be RNA, and it can be single-stranded or it can be double-stranded. And in this image, we just see these subcategories and then all these different types of viruses that form these categories. I'm just going to briefly explain the replication process. I won't go into too much detail, but the general concept is that first a virus attaches to the cell. So a virus has these proteins on its surface, and it can use these to enter and attach to a cell. Once a virus has entered the cell, it then locates the nucleus, and it releases its genetic material into the nucleus. The nucleus then replicates this genetic material, and the cell's organelles then reconstruct viruses. And then this new virus that can then break out of the cell. So I find this concept really cool, that a virus uses its host cell to make new viruses. unless the viruses have a really large variety of morphologies. So they can have like a isokedral structure like in A, or an ovoid structure like we see in the bottom left in K, or even a head and tail structure like in J. Now moving on to space modules. So space modules provide an excellent condition for Earth microbes to spread and grow for numerous different reasons. So firstly, astronauts are exposed for a very long time to viruses. And this means that the chance of infection is increased because they are so long exposed to the same viruses. Astronauts also live in these compact and confined living space, and this also increases the chance of infection through contact or through droplet infection. The air is recycled and filtered, so it's not like on Earth you can open a window and get fresh air. It's the same air revolving around in these space stations. There is an altered oxygen and carbon dioxide ratio. We have controlled temperatures and controlled humidity, and this constant environment can provide a great environment for viruses to thrive and grow. There's also the effect of microgravity, and I will talk more in detail about this later. And then also ionizing radiation and UV radiation, which again, I'll talk more about in detail later, but these all affect both the host, the astronauts, and the viruses. So the paper discussed viruses in the microbiome of the ISS surfaces. So I'm sure you all know, but the ISS is the International Space Station, and it's the largest modular space station and the largest satellite currently in low Earth's orbit. And a lot of scientific research is conducted on the ISS. So this study was published in 2019 by Moore, and they did a shotgun metagenomic sequencing of environmental surface swabs on the ISS. So this approach allows for all genes in the sample to be analyzed and determine what microbes are in the sample. And this approach works really well, even if there are very small amounts of organism or gene present. And how exactly this metagenomic sequencing works is it takes a sample and extracts the DNA, and then it fragments the DNA, and these fragments can then be analyzed. So in orange, we can see that 95% of the viruses, these reads originate from bacteriophages. And this means, so bacteriophage is a virus that infects and replicates only in bacterial cells. And this is, for example, dangerous in our gut. So we have a lot of bacterial cells in our gut doing very great things for us, and viruses could infect these bacterial cells and alter the human microbiome, so this can be dangerous. In blue, we see that about 4% of the reads are derived from animal and human viruses. So they then compiled this data and these reads from these 72 different virus genera, and they distributed these into 21 different families. So underlined, we can see all the human viruses, and we can see there's high readouts for the papillomavirus and herpes virus. But what we also see is a relatively low abundance of viral gene readouts. And the paper mentioned a few explanations for this. So firstly, viral genomes are underrepresented in the genomic databases that are used to assign these sequencing. So there's like the metagenomic analysis uses these databases, and the viral genomes are really widely underrepresented. So it could be that a large portion of these remain unidentified. And it's also worth noting that the metagenomic analysis was performed only on a pooled subset of environmental samples. So it could be that some viruses were missed during the analysis. But what we can take away from these results is that pathogenic viruses were present in quite a low abundance and were unlikely to cause significant health problems on short-term missions, but the impact on long-term missions remains unknown. Now, moving on to the impact of space-related stresses on viral stability. So the 4 stresses the paper discusses include UV radiation, ionizing radiation, humidity, and microgravity. So starting with UV radiation. So UV radiation comes from the sun, and our ozone layer absorbs most of it. In fact, it absorbs 98% of the UV light. It absorbs all of the UVC and most of the UVB we can see, but still a lot of the UVA can reach the Earth's surface. Now, in the skin, in the schematic here, we can see the different layers, the epidermis, the dermis, and the subcutaneous tissue. And we can see that UVA reaches all the way into the dermis, whereas UVB only reaches into the epidermis. And UV can cause DNA damage. So it causes oxidative DNA damage, causes damage to the DNA bases, basically. And if there's too much of this damage or it's not able to be repaired, this can lead to cancer. And in space, we don't have this protective ozone layer, so there are much higher levels of radiation. So these astronauts in space are exposed to much higher levels of radiation than on Earth. So they have much higher chances of getting DNA damage. And UV light also can cause damages to viruses. So it also can damage the nucleic acid and can damage viral surface proteins. So this is a good thing because viruses can also be inactivated by UV light and killed. And in particular, UVC causes inactivation of viruses. But from a recent study by Virology in 2011, they found out that different viruses require different doses of UV to become inactive. So some are more resistant than others. And Tseng and Lee found in 2007 that overall DNA viruses tend to be more resistant than RNA viruses, and those with double-stranded genomes are more resistant to UV radiation than those with single-stranded genomes. And UV can also reactivate dormant viruses. So I find this really interesting, but UV can actually cause dormant viruses to become reactive again in the body. So dormant viruses, this just means that sometimes viruses hide in the cells and they don't replicate or reproduce. They're just simply there, not doing anything. And in certain stress situations, or for example, if there's UV light, these viruses can become active again and infectious. And the mechanism of this reactivation is related to how UV radiation actually suppresses the immune system. So there was a study in 2008 by Rana, and what they found was that UV radiation activates regulatory T cells. So these are a type of cell, a type of immune cell, and they produce immunosuppressive signals. And this inhibits the immune system. So viruses can then exploit this compromised immune system and provoke disease. And we have seen this already in space of viruses that were dormant becoming reactivated and causing illness amongst the astronauts. Now moving on to ionizing radiation. So ionizing radiation is a type of energy released from atoms that travel in the form of electromagnetic waves. So this can be gamma or X-rays, or it can be traveling in the form of particles like neutron, beta, or alpha particles. There are naturally occurring radioactive materials that are found in the air or in rocks. And cosmic rays also contain high quantities of radiation. So just like with UV light, ionizing radiation can also cause DNA damage. So it can be a direct DNA damage, or it can be indirect, forming these ROS, which is reactive oxygen species. And these reactive oxygen species then damage nucleic acid, proteins, and lipids. And they can also cause strand breaks and base oxidation. So this just means that it's damaging our DNA. And again, if this happens too much and we can't repair this, then this can also lead to cancer. As we see here, we have these mutations which can lead to cancer. So Long-term exposure to a low-intensity ionizing radiation increases the chance of developing cancer, especially in the skin and in the lungs. Much like with UV radiation, gamma and X-rays also have been shown to have immunosuppressive effects that lead to the reactivation of herpes and rabies viruses and an increase in illness severity. So yeah, there's been studies that show that radiation can induce the activation of lysogenic viruses via the lactic cycle of herpes virus. And this was shown by a study by Mitte in 2018. Now, humidity is also something that affects viral stability. So humidity, like I said, is a predictor of viral stability, and it actually depends on the presence of a lipid envelope. So viruses with a lipid envelope were shown to be more stable at lower relative humidities, like around 30%, and those without an envelope and simply a protein shell were more stable at higher relative humidities, like around 80%. In this graph here from a study by Ma in 2019, we can see here the percent of viable cells and the humidity. So an increase in humidity leads to a decrease in viability of viruses. So more humidity is basically bad for viruses. Not only that, but a high humidity also thickens human mucosa. So mucosa is produced by goblet cells, which are specialized epithelial cells. And this lines, for example, the airways. And it produces this mucus to trap viruses and pathogens. So if we have a higher humidity, then we have more of this mucus, and it means that this could be trapping dangerous viruses and also bacteria entering our body. So we could use this in space, for example. If we can control the environment and have a slightly more humid environment, this could reduce virus transmission. Now, microgravity. So microgravity is something we can't really replicate on Earth yet. So there is really not that much known about the effects of microgravity on human cells and also viruses. But in space, we have these varying G-forces during space travel. So when launching, we have these really high G-forces, and we have high G-forces when there's these orbital transfers. So when the spacecraft moves from one circular orbit to another if you're in an elliptical orbit. And then once you're in orbit, you have this microgravity environment. In a paper by Alvarez in 2019, they found that in a simulated microgravity environment, the epithelial cells had a decreased function. So epithelial cells, the ones they studied were cells lining the gut, and they found that in this microgravity, they had an impaired function. And this has implications for gastrointestinal homeostasis of astronauts in space. So if these cells are impaired and they're not working properly, it can mean that they have an increased permeability of this epithelial barrier, which means that it could be easier for viruses to enter the body through the gut lining. So hopefully in the future, we will be able to recreate this microgravity on Earth so we can further study this in tests to see how it affects the cells of our body and how it affects viruses and also bacteria. So how can we control viruses in space? So one thing, like I mentioned earlier, that NASA did was a pre-flight quarantine. So they're really trying to stop viruses from entering space and infecting astronauts already on the ISS, for example. Certain materials also have antimicrobial properties like copper and silver. So you could coat the surfaces with these metals to try and prevent the spread of viruses and bacteria. You can also sterilize the spacecraft before it gets sent to space. So we learned earlier that UV light can inactivate viruses. So we could use UV light to sterilize the spacecraft. And we can use air filtration to try and remove any viruses in the air. So they now use a high efficiency particulate air filter called HEPA filters. And this really tries to remove any tiny viruses in the air. So lastly, as I come to the end of this paper, let me just briefly mention some of the strengths, weaknesses, and future outlook. So I thought this paper was really well written and it compiled data from numerous different studies. I found it really interesting to read about the effects of ionizing radiation and UV radiation on viruses and humans, because in space there's not this protective ozone layer like we have on Earth. So the astronauts are exposed to much higher amounts of radiation. So I think it's really important that we study and understand this, not only to understand the effects it has on viruses, but the danger that it causes to astronauts. I did, however, find that many of the studies were quite old and they could have included more recent findings. One thing they didn't do is discuss which viruses are the most harmful and which pose the most threat in space. And I would have found it nice if they had discussed this, because I think it's really important that we understand which viruses are really dangerous to us and which pose this threat if we are to send astronauts on long-term missions to Mars. Because on these long-term missions, they only can take a limited medication and they can't simply come back to Earth for medical help. So they're really on their own out there. So I'm just going to end with some ideas that I thought could have been included in the paper. So maybe you know already that we have the same number of bacterial cells as human cells in our body, so around 37 trillion. But maybe you don't know, but we have 10 times as many virus particles. So we have 380 trillion viruses that make up the human virome. And interestingly, not all viruses are dangerous. Some are actually beneficial for the host. And some viruses kill bacteria, and others actually protect against more dangerous viruses. So here in this image, you can see, let me just get the laser pointer again. So you can see here is the bacteriophage. And this bacteriophage I mentioned earlier is simply a virus that can replicate in only bacterial cells. And here we see the bacteria cell in red. So these viruses can infect and kill bacteria. So some bacteria that are more dangerous for us could be killed by these viruses and actually help and protect us. The question then remains, what happens if UV and ionizing radiation actually suppress and kill these viruses in our body? Is this going to then have negative consequences on us and the astronauts in space? So I think an important question like this can be researched further. So what happens if this radiation is killing these beneficial viruses in our body? And I also discussed earlier that radiation can cause reactivation of viruses. So reactivation of dormant viruses already in the body. So one thing we could do before sending astronauts to space is screen them for these more common viruses like HIV and herpes, and see if these viruses are in the host, and then see if they're able to be reactivated. And if they are, we could send them with the relevant medication. And I think we need much further research into this viral reactivation in the spaceflight environment, because it's really dangerous. We've seen this before in space, of viruses being reactivated and infecting the astronauts and making them ill. So that brings me to the end of my presentation. Here are just some references. I just want to say thank you for listening, and I look forward to reading your questions.